everyone, my name is Isabella. I'm a student writing intern at the College of Letters and Science, and today I talked with two scientists at UW-Madison about their research with zebrafish, and we also looked at some imaging in their lab.
Hi, I'm Drerup. I am an assistant professor in the Department of Integrative Biology in the College of Letters and Sciences at UW-Madison.
And I'm David Ehrlich. I too am an assistant professor here in Integrative Biology.
Why do many scientists study zebrafish in their labs?
We use zebrafish for a lot of reasons for our work. These animals are fertilized externally, so we have access to the eggs from the one-cell stage. This means we can manipulate their genomes pretty effectively, creating overexpression and knockout models of different disorders to try to understand neuronal cell biology. And in particular, after we make these modifications, we can use in vivo imaging in order to track cells and even things inside individual cells in real time using fluorescent markers. This is because these animals develop very rapidly, and we have the tools now to manipulate their genomes, and also the larvae are transparent, meaning we can put the whole animal under a microscope for some really effective imaging in an intact neural circuit.
For a lot of scientists, the fact that larval zebrafish are vertebrates means that many of the biological processes are conserved throughout evolution from the larval zebrafish up to humans even. So we can study biological processes that are involved in diseases or perform basic science research to elucidate some general mechanisms. So for my work, we try to understand how the nervous system processes sensations and generates movements and actions. And larval zebrafish, even though they are, smaller than a grain of rice, they use many of the same processes to generate movements and to interact with their environment that we do. So we can study things in a simpler way and have more control over the experiment, but still study some of the same basic processes.
The beautiful thing about this type of imaging setup, as well as this organism, is we can do in vivo imaging of a live animal. So this is the head of a zebrafish larvae at 4 days post-fertilization. So going from a one-cell stage animal to a freely swimming, freely feeding larvae happens in just 4 days. So you can see here the head with the eye and the brain. And this animal is expressing a transgene that makes their neurons fluoresce green by encoding green fluorescent protein under a neuron-specific promoter. So you can see here that this live animal was just embedded and imaged, allowing you to see the beautiful structure of the nervous system and even individual cells. All of these bright dots are individual neurons in the animal. Not only can we look at the structure of the nervous system and the location of different cells, we can also zoom in and look inside a neuron and ask what happens to the fundamental core intracellular structures that make up this animal's nervous system. And we can use this kind of imaging in order to understand not only the structure of the neurons, but also how these intracellular processes, like mitochondrial transport, impact the structure and the function of the nervous system.
So the first part of a project might look something like this. We've got here an apparatus where we take high-speed video of larval zebrafish as they're swimming and as they're responding to stimuli that we create and present them. In this case, a graduate student in the lab, Nick, was filming larval zebrafish. You can see them here in the top-down view and here from the side. So what Nick is doing is tracking how they're contracting their muscles and moving their bodies in 3 dimensions to try to understand what signals are coming out of the nervous system to tell the muscles when and how to contract. And in this case, what Nick was doing was filming them swimming along as they spontaneously do, and then presenting them with a stimulus called a loom that simulates a predator approaching and getting much larger very quickly. And so what you'll see when I hit play is the fish in slow motion, bending their tails back and forth. Again, here in the top-down view, you can see them distributed throughout the depth of water. And now the fish are starting to respond to that looming stimulus, and you'll see them swimming away from the stimulus here and making rapid escape maneuvers.
What are you using zebrafish to research?
We study parts of the brain that are involved in learning new movements and controlling actions, including parts of the brainstem and cerebellum. And we try to understand how cells in those parts of the brain allow a fish to turn its sensations into swimming maneuvers, and ultimately use that insight to understand how humans and all animals, all vertebrates, learn to process sensations and generate actions.
My lab is much more focused on cell biology, actually, and it kind of illustrates the many different types of science you can do with zebrafish because of their many attributes. So we like to use zebrafish to try to understand how neurons, individual cells in the brain, are both formed and then maintain their very complicated architecture over the life of the animal. So neurons rely on a lot of intracellular processes in order to both find their connecting partners and then to maintain those connections throughout the life of the animal. In humans, disruptions to a lot of these processes are contributing to neurodegenerative diseases, for example. So we hope that by studying how neurons are both born, form their connections, and then maintain those connections, will give us some insight into how to keep neurons healthy over the life of a fish, but also these same processes are at play in the brain of a human as well.